Okay, so welcome to this next video in which we are discussing the mammalian circadian timing system. Okay, so we're in the process of discussing how visual information coming from the retina is going to entrain the circadian clock that is within the suprachiasmatic nuclei. Okay, so we want to see how we keep the suprachiasmatic nuclei neurons, we keep all of their circadian oscillators in tune with the light-dark cycle. Okay, and we've seen that on the retina you have these photoreceptive retinal ganglion cells which have the pigment melanopsin in them. Okay, and this can detect light, which is very rare for retinal ganglion cells. Usually they don't directly detect light. And these photoreceptive ganglion cells send their axons into the optic nerves, and then they move from the optic nerve or via the optic chiasm up into the suprachiasmatic nuclei, and that movement of the axons up from the optic nerve or the optic chiasm into the suprachiasmatic nuclei is known as the retinohypothalamic tract. And then what they're going to do is synapse on suprachiasmatic nuclei neurons, and they're going to basically entrain them. And we want to see how, if a suprachiasmatic nuclei neuron is out of sync basically with the light-dark cycle, how this information coming from the retina is going to help get it back in sync with the light-dark cycle. Okay, so we've discussed that these neurons from the retina are going to release two signaling molecules onto the suprachiasmatic nuclei neurons. One of them is going to be glutamate, the other is going to be PACAP. We'll come back to PACAP. We're going to talk about glutamate first. So we're currently discussing the ionotropic glutamate receptors, which are the main receptors that you have on the suprachiasmatic nuclei neurons, which are going to detect the glutamate. We've discussed that ionotropic glutamate receptors consist of tetramers. Okay, so you have 4 polypeptides joined together, and that each of those subunits is going to look like so, with this same basic structure here. Okay, we now want to discuss the 3 different families of ionotropic glutamate receptors, and we're going to begin with the AMPA receptors. Okay, so we're going to start off with the subunits that make up AMPA receptors. Okay, so what would be really simple is if there was just one subunit known as the AMPA receptor subunit, and all you had to do is make 4 AMPA receptor subunits, bind them all together into a tetramer, and there was your AMPA receptor. But it's not that simple. Instead, there are 4 different subunits that you can use to build an AMPA receptor. Okay, so we're going to start with AMPA receptors here. Okay, so these 4 different subunits are called GluA1, okay, GluA2, GluA3, and GluA4. Okay, and they all have old names. These are the names that they've been renamed with. They've been rebranded with these new names because these new names are very simple. Okay, but they have old names. GluA1 used to be called GluR1. Okay, and if you read older papers on glutamate receptors, you will definitely see this nomenclature. Okay, GluA2 used to be called GluR2. GluA3 used to be called GluR3, and GluA4 used to be called GluR4. Okay, so that's quite nice and simple. It's not too difficult to remember that. But as we'll see, this notation for the subunits of the AMPA receptors, by naming them GluR, that continues on through to the subunits of the kainate receptors, and if you don't understand what I mean by that, you will later. Okay, right. So, all of these 4 subunits then can be used to make a quarter of an AMPA receptor. They all have the same basic structure. They have an amino terminal domain followed by this S1 domain, which is going to participate in the formation of the ligand binding domain. Then we'll have the M1 domain, then the reentrant loop, which is M2, then M3, then S2, and then M4. Okay, but all of them will have slightly different sequences of amino acids. So although they have this same sort of core structure, they aren't identical. They are different proteins fundamentally, but they can all be used to make a quarter of an AMPA receptor. Now, how then do we actually tetramerize these together? These are the 4 subunits, but of course our AMPA receptors consist of 4 subunits all put together. And the question then is, how do we actually get 4 subunits together to make that? I.e., do we use 4 of the identical subunits, or can we mix and match a little bit? Well, basically, there are 3 known homotetramers, which means these receptors where you have 4 identical subunits. So the GluA1, okay, the GluA2, and the GluA4 can all form homotetramers, which means you can take, for instance, if we use GluA1 as the example, you can take 4 GluA1 subunits, tetramerize them together, and you then get a GluA1 homotetramer. So homo means same, tetramer means 4-membered structure. Okay, so it's a 4-membered structure where all the subunits are the same, basically. Okay, and you can likewise do the same thing for GluA2 and GluA4, and those make you AMPA receptors then. Okay, but you can also mix and match to an extent. Okay, you can also make tetramers where you don't have just one type of subunit within the tetramer. Okay, now, these are called hetero tetramers. Hetero means different, and then tetramer again means 4-membered structure, so the 4 subunits that you're going to make the tetramer out of are going to be different. Now, it doesn't quite go wild. You can't, for instance, take 1 of each type of subunits and stick them together and make an AMPA receptor. We haven't found that in nature. Instead, you can make things known as di-heterotetramers, and I regret now putting that as a capital letter, but never mind. Okay, so what this means is that di means 2, so you're going to use 2 different subunits to make up your full tetramer, and those are the only AMPA receptor heterotetramers we've found, i.e. heterotetramers which consist of 2 different types of subunit, so not 3 and certainly not 4. Okay, so let me give you some examples of di-heterotetramers which we know exist. So basically, we have 3 main di-heterotetramers, so there is GluA1 paired with GluA2. Okay, so what this means is that you've got a tetramer which contains some GluA1 subunits and some GluA2 subunits. And I don't know whether it's known, and I don't think it is known, what the stoichiometry of these are, i.e. whether you use 1 GluA1 subunit and 3 GluA2 subunits, or 2 GluA1 subunits and 2 GluA2 subunits, or 3 GluA1 subunits and 1 GluA2 subunit. And I certainly don't know, for instance, if you use the 2 and 2, how you arrange them relative to each other, i.e. Because there would be 2 plausible arrangements. You could have the 2 GluA1s next to each other, okay? For instance, this one could be an A1, this one could be an A1, and then these 2 could be A2s, or you could have them diagonally opposite each other, so you swap this one with this one, and then you've got them nicely diagonally opposite each other. I certainly don't think that's known. Okay, right. What we do know is that GluA1 and GluA2 can be found together in a di-heterotetramer, basically. So you can find AMPA receptors which have both GluA1 subunits and GluA2 subunits within them. Okay, and then other di-heterotetramers you can then find are the other 2, GluA3 and GluA4, also paired with GluA2. So what do I mean by this? I mean you can also find GluA2 paired with GluA3, okay, and also GluA2 paired with GluA4. So basically you can pair GluA2 with any of the others, and those are the di-heterotetramers that we find within nature. Okay, so those are the different types of AMPA receptors. We have these homotetramers and then these di-heterotetramers. Okay, what I now want to explain to you is when glutamate acts on these AMPA receptors, what is going to happen? What is the pore actually permeable to? Okay, right, so let's go over the page. So, basically, the pore is going to be permeable to sodium and potassium ions, always. Okay, so when glutamate binds to the ligand binding domains of the 4 receptor subunits that are making up the entire AMPA receptor, it will open a pore, and this poor will be permeable to sodium and potassium. Okay, sometimes it's also permeable to calcium, and this depends on which of the AMPA receptors you are talking about, because we've seen many different types of AMPA receptors now. We've seen these 3 homotetramers, which are all AMPA receptors, and we've seen these 3 Hetero Tetramers, which are all AMPA receptors. In order to be permeable to calcium, you need to not, I repeat, not have a Glu-A2 subunit within you. If you have a Glu-A2 subunit within you, that abolishes your permeability to calcium. Okay, so that's a pretty steep requirement, considering that 4 of our AMPA receptors that we've seen here do have Glu-A2 in. So the Glu-A1 does not have Glu-A2 in, and the Glu-A4 homotetramer doesn't have Glu-A2 in. But certainly the Glu-A2 homotetramer does have Glu-A2 in, and all of these diheterotetramers at least have 1 Glu-A2 subunit in. So none of these ones are going to be permeable to calcium. The ones that I've highlighted in red are going to be permeable to calcium. So you're permeable to calcium ions, at least slightly, if you do not have a Glu-A2 subunit in. So for this, you need no Glu-A2. Okay, now I want to just discuss what the effect of opening an ion channel that's going to be permeable to sodium, potassium, and potentially calcium is going to have on the electrical potential difference across the cell membrane. Okay, right. So firstly, let's discuss electrical potential difference, actually. And for this, we need to firstly discuss electrical potential. Okay, so electrical potential is this concept from physics. It's a mathematical model to help us understand the world around us. Okay, and this is the model. What you do is you go to every point in three-dimensional space and you ascribe it a real number. So for instance, we'll highlight this point in turquoise here. This point, as with all other points in three-dimensional space, this is my attempt to draw a three-dimensional graph, will be given a real number. Okay, this real number can be positive, it can be 0, it can be negative, it can be an integer, it can be a rational, it can be an irrational, it can be whichever number you want. So you ascribe to this a real number, and that real number is going to be called the electrical potential. Okay, so I'll call the electrical potential big E. Okay, right, so every point in three-dimensional space is given this number. Now, this means that in principle, I can have a little man shown here with a special machine here, okay, and this special machine is going to measure electrical potential. And in principle, this little man can go and stand wherever he wants in three-dimensional space, and he can measure the electrical potential at that point. Okay, right, so now away from the physics, and let's go back to biology. Okay, so let's have a cell here. Basically, all the points inside the cell, so all the points within the intracellular compartment, which I'm now highlighting in orange here, all of those points will have the same electrical potential because they're all going to be in electrical equilibrium with one another. So all of the points inside the cell are going to roughly have the same electrical potential. So what that means is if this little man comes into the cell with this machine and walks around, he can stand wherever he likes and he will get the same number on his screen. Okay, now let's call that number that will be on his screen the electrical potential, capital E, intracellularly, so capital I for intracellularly. So the big E is for electrical potential, and the capital, the subscript I is for intracellularly. Okay, so this stands for electrical potential. Okay, right. So all of the points of the intracellular fluid have the same electrical potential. Now let's move into the extracellular fluid. So I'll highlight this in pink. Okay, again, all the points in the extracellular fluid are going to be in electrical equilibrium with one another and therefore going to have roughly the same electrical potential. So if the little man goes and stands in the extracellular fluid now, he can walk around and stand in many different places and he's going to get the same number showing on his screen, and we'll call that capital E again for electrical potential, and we'll subscript it capital E as well for extracellularly. Okay, so the intracellular fluid has a certain electrical potential. The extracellular fluid also has a certain electrical potential. The question now is, are these two numbers the same? And the answer is no, not usually across a cell membrane. Okay, they do not now intracellular fluid and the extracellular fluid do not need to be in electrical equilibrium because they have the cell membrane separating them. Okay, so there is now the concept of there being an electrical potential difference across the cell, okay, which is the concept of a difference in electrical potential, okay, and electrical potential differences are also called voltages. Okay, now when people talk about the electrical potential difference across the cell membrane or the voltage across the cell membrane, what they really mean, what they really strictly speaking should say is the electrical potential difference or the voltage from extracellular to intracellular. They should in principle give you a direction. They should say from one place to the other place. Okay, so from extracellular to intracellular. Now, what then does this mean? This means if the little man started in the extracellular fluid, okay, so he started over here, let's say, then the number on his screen would be the electrical potential extracellularly. It would be this number here. Okay, if he then moves from extracellular to intracellular, so he follows my trajectory here and ends up in the intracellular fluid, okay, and then the number on his screen changes, it will change to this number here. The question now is, how much does that number on his screen change when he moves from extracellular to intracellular? Okay, so what is the answer to that? Well, the number after he's moved is the electrical potential intracellularly, and the number before he moves is the electrical potential extracellularly. So all we need to do to find how much the number changes when you move from extracellular to intracellular is subtract the later number, the number after the move, from the number before the move. And this will give us the electrical potential difference from extracellular to intracellular. However, people assume that you realize that we always mean from extracellular to intracellular. Okay, we're always talking about from going into the cell, okay, from the outside into the cytoplasm, okay, because usually that's what scientists have to do. They have to stick the electrode into the cell. Okay, right, so we always talk about going from extracellular to intracellular. So people will often just say the electrical potential difference across the cell membrane or the voltage across the cell membrane, but this is strictly speaking what they mean. Okay, right, so normally this number is kept at around negative 65 Oh, and I never actually told you about the units of electrical potential. So let's firstly talk about the units of electrical potential and then we'll come back to this. Okay, so the units of electrical potential are volts. Okay, so every place in three-dimensional space has this number, which is a real number, and the units are volts. Then if we ask what's going to be the units of electrical potential difference, okay, well we are taking one number measured in volts away from, well we're taking one number and we're subtracting off another number that's measured in volts. So we're going to end up with another number that's going to be in volts. So the electrical potential difference across the cell membrane is also in volts, basically. Okay, so the electrical potential difference across the cell membrane then is usually kept as around negative 65 millivolts. Now a milli just means a thousandth of a volt. Okay, so if you like, you can rewrite this as negative 0.065 volts. Okay, so what then does this mean? This means that if the little man moves from extracellular to intracellular, the electrical potential difference that is shown on the screen of his machine goes down. Okay, that's the negative bit, and it goes down by 65 millivolts. So the intracellular compartment's electrical potential is lower than the extracellular compartment's electrical potential, and it's lower by 65 millivolts. Okay, right, so now back to the AMPA receptors and what's going to happen when we open our AMPA receptor. So here is the cell membrane, and we'll draw our AMPA receptor in here. Okay, and it's a little bit squashed. I don't quite know why I've drawn it all the way over here, but never mind. Okay, so here's our AMPA receptor. Remember, it's a tetramer of 4 separate subunits, like so. Okay, and now what's going to happen is glutamate molecules are going to come from the axons of those photoreceptive retinal ganglion cells, and they're going to release glutamate onto our suprachiasmatic nuclei neuron. And our suprachiasmatic nuclei neuron is going to have receptors of all three kinds on it. So it's going to have AMPA receptors, kainate receptors, and NMDA receptors. Okay, so we're going to talk about AMPA receptors first, and what the binding of glutamate and the opening of this channel is going to do to the suprachiasmatic nuclei neuron. Okay, right. So in comes the glutamate then. So glutamate will bind to the 4 ligand binding domains on the extracellular side of the membrane. Okay, so each one of the subunits, remember, will have a ligand binding domain. Glutamate will bind, so you'll get 4 glutamates binding in total. So I might just show these glutamates as orange dots maybe. 1, 2, 3, 4. This will now cause the AMPA receptor to open. Okay, and when it opens, it's permeable to sodium and potassium definitely. Okay, and we'll come to calcium later. We'll do that last, because only a few of them are going to actually be permeable to calcium. Okay, right. So we now need to work out which direction are sodium ions and potassium ions going to have a net movement within. Okay, so to do this, we need to know the concentration gradients of sodium and potassium across the cell membrane. So we'll start with sodium. The concentration of sodium extracellularly is around 145 millimolar, whilst the concentration of sodium intracellularly is around 12 millimolar. The concentration of potassium extracellularly is around 4 millimolar, whilst the concentration of potassium intracellularly is around 155 millimolar. Okay, so now let's work out which direction sodium and potassium are going to move. The other piece of information we need is the electrical potential difference from extracellular to intracellular, okay, which we'll assume is around negative 65 millivolts. So we'll assume that this is negative 65. So when you move from extracellular to intracellular, you go down in electrical potential by 65 millivolts. Okay, so we've now opened this pore through the middle of the AMPA receptor. Okay, so sodium ions. Let's begin with sodium ions. So at present we have a 12-fold concentration gradient of sodium favouring the movement of sodium into the cell, simply because a sodium ion is 12 times more likely to hit the pore from the outside and go in than it is to hit the pore from the inside and go out. Okay, so the concentration gradient is going to favour the movement of sodium into the cell. Meanwhile, we have to consider the electrical potential difference across the cell membrane. Sodium is a positively charged ion. Positively charged ions want to go where the electrical potential is lower, okay, which currently is the intracellular compartment. The electrical potential of the intracellular compartment is currently 65 millivolts lower than the electrical potential of the extracellular compartment. So at the moment, both the electrical gradient and the concentration gradient are favouring the movement of sodium into the cell. So that means they're both in agreement, and you're going to get a net movement of sodium into the cell through the AMPA receptor, of course. So I've drawn the arrows that it's going through the membrane, but really, of course, it will be going through the pore of the AMPA receptor. Okay, now let's turn our attention to potassium. Potassium then, you've got around a 40-fold gradient of potassium favouring the movement of potassium out of the cell. However, potassium is also a positively charged ion, so it too is going to want to stay in the intracellular compartment because the intracellular compartment has the lower electrical potential. So the electrical gradient says go in, the concentration gradient says go out. The question now is which wins, which is stronger? Okay, so you've got the concentration gradient saying this, you've got the electrical gradient saying this, and I've given it away which is stronger now. The concentration gradient wins, basically. It beats the electrical potential difference of negative 65 millivolts. In actual fact, you'd have to take the electrical potential difference across the cell membrane down to around negative 85 millivolts, the Nernst potential for potassium, until you'd get absolutely no net movement of potassium across the cell membrane, because then the electrical gradient was strong enough to completely match the concentration gradient, and you then get no net movement of potassium. And then if you went beyond there, let's say to negative 95 millivolts, then you'd actually get potassium coming into the cell, because the electrical gradient would now be stronger than the concentration gradient. Okay, but we're at negative 65 millivolts, so the concentration gradient is stronger than the electrical gradient. Okay, so you are going to get a net movement of potassium out, but of course it's going to be hugely weakened by the electrical gradient. So this in purple now is the net movement of potassium out. So what this means then is that you're going to get more sodium moving into the cell than you're going to get potassium moving out of the cell, which means that you've now got a net movement of positive charge into the cell, because both sodium ions and potassium ions carry a positive charge. So we're moving this much positive charge into the cell in the form of sodium, we're moving this much positive charge out of the cell in the form of potassium. That means that we're going to have a net movement of positive charge into the cell, because the movement of sodium beats the movement of potassium. Okay, so we're bringing in positive charge into the cell. Okay, now what effect is this going to have on the electrical potential difference across the cell membrane? Okay, so basically when you have a movement of positive charge like this, it's known as a depolarizing current. Okay, so let me explain that term. So a current just means a movement of charged particles. This is certainly a movement of charged particles. Depolarizing current means that it's going to depolarize the electrical potential difference across the cell membrane. Now there's another name that's often used for a depolarizing current. It's called an excitatory postsynaptic current, or an EPSC. So this stands for an excitatory, postsynaptic current. Excitatory because it's going to make it more likely that the neuron that is postsynaptic will fire an action potential. And then current again because we're working with a movement of charged particles. Okay, and postsynaptic then just because it's the postsynaptic neuron. Okay, right. So what does this do then? Well, it's going to depolarize and excite the postsynaptic neuron, is what we've gathered from the name of it. Okay, but why? Well, when you bring positive charge from the extracellular fluid into the intracellular fluid, the effect that this is going to have on the electrical potential intracellularly and the electrical potential extracellularly is that you're going to raise the electrical potential intracellularly, and the reason is wherever you dump positive charge, that raises the electrical potential of that area. So we are taking positive charge from the extracellular fluid and dumping it into the intracellular fluid. That means we're going to raise the electrical potential of the intracellular fluid because we're adding in additional positive charge there. Meanwhile, because we're grabbing the positive charge from the extracellular fluid, we are taking positive charge out of the extracellular fluid. Okay, now that positive charge that was present in the extracellular fluid, that would have been boosting the electrical potential of the extracellular fluid. Okay, so when we remove that positive charge of from the extracellular compartment, we will be reducing the electrical potential extracellularly. Okay, so we're going to be raising the electrical potential intracellularly, whilst we're going to be reducing the electrical potential extracellularly. The question now is, what effect is that going to have on the electrical potential difference from extracellular to intracellular? Okay, so let's firstly try and do this just through intuition, and then we'll go to the formulas. Okay, so initially, the electrical potential extracellularly was 65 millivolts higher than the electrical potential intracellularly. Okay, so if I draw you a little graph, okay, and believe it or not, a graph actually helps in this case. Where should I put this? I'll put this down here. Okay, so if we have, what am I doing there? Why am I doing that? I don't want it slanted like that. Okay, so if we have electrical potential on the y-axis here, then basically what we've got is we've got a high electrical potential here. Let's say this is the electrical potential extracellularly, and let's say this is the electrical potential intracellularly. Okay, so the electrical potential intracellularly is lower than the electrical potential extracellularly, i.e. you step down when you move from extracellular to intracellular. Okay, and this step down at the moment is negative 65 millivolts. Okay, now we are lowering the electrical potential extracellularly. Okay, so this number is going to go down like so, and we are raising the electrical potential intracellularly. So this number here is going to come up. So hopefully you can now see that the drop that you're going to suffer is going to be reduced basically. Okay, so the electrical potential difference from extracellular to intracellular is going to become less negative. Okay, and the name for the electrical potential difference across the cell membrane becoming less negative is to call it a depolarization. We say that the electrical potential difference across the cell membrane has become depolarized. Okay, right. To see that in terms of the maths, here is our formula for electrical potential difference from extracellular to intracellular in terms of the electrical potential intracellularly and the electrical potential extracellularly. Okay, we are raising the electrical potential intracellularly, we're raising this number here, and we're reducing this number here. Okay, so we're raising the first number and reducing the number we're subtracting off, so clearly this number is going to become less negative, i.e. we're going to get a depolarization. So, it's going to become less negative, so it might go up to something like negative 55 or higher maybe. Okay, so that's the effect then of opening an AMPA receptor on the electrical potential difference across the cell membrane. You get this depolarizing current which will cause a depolarization in the electrical potential difference across the cell membrane. And there is a fancy name for this depolarization of the electrical potential difference across the cell membrane. This is known as an EPSP, okay, which stands for excitatory, that's the E, okay, and then postsynaptic again, just because we're on the postsynaptic neuron, and then the final P this time is for potential. So the name for this depolarization in the electrical potential difference across the cell membrane due to the opening of these AMPA receptors is the excitatory postsynaptic potential. Now, the other thing I should stress is that obviously you're not just going to have one AMPA receptor on your postsynaptic neuron, you'll have loads of them, and they'll all be opening in response to glutamate, they'll all be letting in these little depolarizing currents, and they'll be adding together to make a total excitatory postsynaptic current, and that will give you the total excitatory postsynaptic potential. Okay, right. Now, the final thing I just want to discuss is if we've got an AMPA receptor which has no GluA2 subunits within it, then of course we'll have an AMPA receptor that is permeable to calcium ions. Okay, so we need to add in calcium ions and which direction they're going to move in on this picture here. So for this, we need to know the extracellular and intracellular concentrations of calcium. So calcium extracellularly is around 1.5 millimolar in concentration, whilst intracellularly it's around 100 nanomolar. Okay, that's a 15,000-fold gradient favouring the movement of calcium into the cell. So we don't even need to think about the electrical potential difference. No electrical potential difference across this membrane that is physiological is ever going to be able to stop the movement of calcium into the cell, okay, when you open a calcium channel. However, in this case, the electrical potential difference is actually in agreement with the concentration gradient, so it's going to make this gradient even stronger, basically. Okay, because calcium is a positively charged ion, it's actually a divalent cation. Okay, so it most definitely wants to be in the compartment with the lower electrical potential, which of course is currently the intracellular compartment. Okay, so both the concentration gradient and the electrical gradient are favouring the movement of calcium into the cell. Okay, now I should not have just drawn that massive great arrow because the amount of calcium that's actually going to move is not going to be that massive. Even though the gradient is massive, you have to bear in mind that the concentration, the actual concentration that you have out here is much smaller than that for sodium. Okay, so the actual probability of an ion going through the channel is lower than for sodium. So you will get a movement of calcium in, but it won't be bigger than the movement of sodium. So that's a little bit misleading. I regret drawing that arrow. I'll sort of cross a little bit of it out. There we go. You will get a movement of calcium into the cell, however. And in the cases for AMPA receptors where they do have this permeability for calcium, which is those AMPA receptors with no GluA2 subunits, that will increase the size of the positive current that's moving into the cell, because we've now got an additional calcium current. Okay, so that will increase the amount of positive charge coming into the cell. So the effect will still be the same, that you're letting in this depolarizing current, okay, into the cytoplasm of the cell, and this will depolarize the electrical potential difference across the cell membrane. Now, we're going to see that the calcium signal is actually going to be extremely important for actually getting the suprachiasmatic neurons, circadian oscillators, in tune with the light-dark cycle. Okay, so we're going to come back to the calcium signal. However, it's not really the calcium signal that comes through AMPA receptors that's important, because the reality is that very few AMPA receptors are actually going to be permeable to calcium. Those that are going to let in very little calcium, and this isn't really the bulk of the calcium that's going to be generated by the glutamate signal. Okay, calcium is going to be very important, and yes, this little bit of calcium that's coming in through AMPA receptors will help, but the main calcium signal is going to come through the opening of the NMDA receptors, which we'll come to in a moment. Okay, so bear in mind that there is this calcium signal, but it's not really that important. We're going to see that the calcium signal is responsible for re-entraining the suprachiasmatic nuclei neuron. But the bulk of the calcium signal comes through the NMDA receptors, and therefore the main effect of these AMPA receptors is to depolarize the electrical potential difference across the cell membrane of the suprachiasmatic nuclei neuron. Okay, and we're going to see that that's essential, that depolarization is essential for the opening of the NMDA receptors later. Okay, right, so we'll call it there for this video, and in the next video we'll turn our attention to the kainate receptors, which shouldn't take us that long because they're very similar to the AMPA receptors.